Biology chapter 19 theory video 2. Now we are going to discuss the tools of the genetic gene technologist in detail. So we discussed about restriction enzymes. To take an example of restriction endonucleases. So these are some enzymes that are found in bacteria, and their function is to recognize and cut the DNA of viruses which attack the bacteria. So these viruses are known as bacteriophages or simply phages. So what they do is they cut up these phage DNA into smaller pieces. So when they are doing this cutting, it is actually cut along the sugar phosphate backbone of the phage DNA, and because they do it within the DNA molecule rather than removing nucleotides from ends, we call it endonucleases, right? And also their function is to restrict the viral infection. So as a result, we call it restriction enzymes. Now, what they do is these restriction enzymes will go and bind to a target site of the DNA that is going to be cut. So then the bacterial DNA also needs to be protected, because it needs not, if it is not protected, it will also be cut. So how it does is by chemical alteration to the bases of DNA, or by simply not having restriction, by not having the target sites, that is by restricting its access to the DNA. So the restriction sites or the target sites are specific sequences of 4 to 6 bases. For example, BAMH1 is an enzyme. This restriction enzyme will cut the DNA when there is a GGA-TC sequence on 5 to 3 strand, and its complementary sequence CCTA-GG on 3 to 5 strand. So because these read the same from both directions, we call it a palindrome. And many of these things, but not all of them, many of the restriction sites are palindromic. Then we speak about sticky ends. So when restriction enzymes cut, it can actually cut straight across the sugar phosphate backbone to give blunt ends, or they can cut it in a staggered fashion. That is, staggered fashion is, sticky ends means there is one short lengths of unpaired bases. So when they form, because they can form hydrogen bonds with complementary sequences on other pieces of DNA, if it is cut with the same restriction enzyme, it is known as a sticky end. So you can identify these cut off pieces by using a complementary gene probe. Gene probe is going to be a length of single stranded DNA with a known base sequence. So when it is used, it will bind with the DNA length having the complementary base sequence. So this will help us to analyze and sort and also store genetic information. Then by using PCR, we can make multiple copies of the strand of DNA. So that will actually be useful in separating and amplifying DNA. We'll be learning about these things in detail later. Then these restriction enzymes would be named using the abbreviations that would indicate their origin. Then there would be Roman numbers at the end to distinguish different enzymes from the same source. Now there would be, for example, ECOR1 comes from Escherichia coli strain RY13 and was the first to be identified from this source. So there would be two ECOR2. So like that we can identify different enzymes from these sources. So we learned actually three key phrases here. Restriction endonucleases or restriction enzyme is an enzyme originally derived from bacteria that would cut DNA molecules, and each type of restriction enzyme would cut only a particular sequence of bases because these enzymes are specific. Then we discussed about bacteriophages. They are a type of virus that infect bacteria. The phages will have double-stranded DNA as their genetic material, so that is what we cut off. Then gene probe is going to be a length of DNA that has a complementary base sequence to another piece of DNA that you are trying to detect. So it is going to be like this. Now here they have given ECOR1, restriction site is 5G AATTC 3, then 35. So as you can see, the site of cutting across is going to be as shown in the third line. I hope this graph makes sense. This table would make sense to you. Please go through it very carefully. If you have any doubts, you can ask. If you do not have any doubts, please go through. Please do question number 2.